Thank you. So it's my pleasure to introduce David Bierbach today for today's seminar. David is a PI at Humboldt University in Berlin with the newly funded Excellence Cluster, the Science of Intelligence at the Technische Universität in Berlin. A lot of David's research focuses on consistent individual differences in labile traits such as behavior. And he works to develop techniques to study behavior in highly standardized ways, including video playbacks, computer animations, and biomimetic robots, which I think we'll be hearing about today. David started his studies at Julius Maximilians University of Würzburg, studying tropical animal ecology, so he has a lot of ties to many of us. He then spent some time as a Hiwi, an intern at the Biota West Africa project and the Frankfurt Zoo, which sounds super interesting and fun. David did his PhD with Dr. Martin Plath at Goethe University in Frankfurt, and he was awarded the best dissertation in life sciences at Goethe University in 2013. He's since moved on to do postdoctoral work with Drs. Jens Kraus and Max Wolf at the Leibniz Institute for Freshwater Ecology and Inland Fisheries, before getting his current position. So today's session will be structured with the talk first, followed by Q&A, of course. But then afterwards, we're going to keep the session open, and if people would like to stay longer, they may do so, and we'll have a bit of a more open, less formal, less structured discussion. Okay, so with that said, I'll invite David to the Zoom stage, and he can tell us about how clonal fish and biomimetic robots help us answer fundamental questions in behavioral research.
Yes, thank you, Anish, for the very kind introduction. And yeah, so welcome to my talk, Constance, and good morning, Constance. I'm David Bierbach from the Humboldt University and from the Cluster of Excellence Science of Intelligence. And I'm very honored and I'm very pleased to present my work on collective behavior to you as the German Center for Collective Behavior Research. And so my initial goal was also to visit your facilities, because I heard so many nice stories of your new facilities for behavioral research that I wanted to visit you. So I hope if I don't do it too bad here, there will be a possibility to also come to Constance and visit you. All right, so today I want to show you two examples of my research, and I will start with a short introduction to the topic. What you see here are more or less fundamental topics of ecology and evolution. We see migration, we see sexual selection action, we see predator-prey interactions, and we see, for example, collective foraging. And all this is linked through behavior. So in a way, behavior is central for ecology and evolution. And our classic view how behavioral phenotypes can develop is depicted here. We have environmental variation and we have genetic variation that are both merged through some developmental processes into an individual behavioral phenotype. Recently, we now know that also epigenetic mechanisms, so modifications to the DNA readability and some random noise affect the behavioral phenotype of an individual. And the fundamental question here that we in Berlin and I specifically ask, what happens if we can reduce the environmental variability? And what happens when we at the same time also reduce the genetic variability? How will the behavioral phenotype look like? Is there behavioral variation among individuals still present when we knock out the two main causes of variation in our animals? And knocking out environmental variability is something that researchers have been doing since years. You see some examples here, a tank system for the laboratory where you can house fish in very standardized environments. You do that also with plants. You do that in aquaculture. So there are many ways how researchers and other people try to minimize environmental variability to animals. It is a little bit more tricky to knock out genetic variability. But here we can use clonal animals to do that. So animals that have the exact same genetic background. And here are some examples listed, for example, the marbled crayfish, but also highly inbred mice that are assumed to be almost clonal. And Kate Laskowski and Max Wolf, together with me and Jens Krause and Carolina Doran, we propose that natural occurring clonal vertebrates are an untapped resource for ecology and evolutionary research. So we think that knocking out the genotypic variability through using these kind of animals is very, very beneficial for our research. And we don't talk here about, more or less the clonal sheep dolly or so, but we talk about natural occurring clones. And in Berlin, we use clonal fish species to do that. And this clonal fish species, the so-called Amazon molly, Poecilia formosa, is a very special organism. It was the first clonal vertebrate that was discovered almost 100 years ago. It lives at the south of the US and the border region to Mexico, and it stems from a hybridization event of two closely related species were mating. So there was a female Poecilia mexicana and a male Poecilia latipina. Both are two common molly species in that region. And they produced a single female offspring that could reproduce clonally. So this clone species derived from one single hybridization event. And these clonal female then copied itself a lot of times. Since 100,000 years now, we find this species in that region. And a very special thing is that they still need the sperm of one of their parental species to develop their eggs. Nothing from the sperm. So no DNA from the sperm is incorporated into the egg, but they still need the sperm to develop the eggs, which is very cool for us in the laboratory because we can really control when the babies are born by adding males to the tank. And in the laboratory, it also works with the ornamental black molly, which make it much more easier to discover the males in the tanks. So in a recent study, we looked how they really do it to control two different genomes that they combine. So they have the genomes of their moms and of their mom and their father. And so genomes of two different species. And this is always hard. We know that hybrids often face some problems having two different genomes in itself. And because both genomes want to get expressed. So there's no fine-tuned mechanism typically in hybrids that control these genomes. And we looked here at the gene expression landscape of the clonal Poecilia formosa and an artificially produced hybrid. So we let Poecilia mexicana, Poecilia latipina individuals mate and produce hybrids with that. They are not clonal. So no one could, has done it to create a clone in the lab, but they have a very similar genetic backup compared to the clonal Amazon molly. And what we find is that the artificial hybrids, they express both genomes at the same time, simultaneously. So here the genomes really fight with each other, while the clonal Poecilia formosa, they either switch on the maternal genome or the paternal genome. So they have a very fine-tuned way to control these different genomes, which is very fascinating and most likely the outcome of these 100,000 years or 500,000 generations of evolution that these clonal fish have experienced. But coming back to the initial question of how much phenotypic variation in behavior is present in clones. So we basically did a twin study on that. So we isolated newborn clones, either really in a single tank, very controlled environment, or in two different treatments, we let them together with their sisters for longer periods, for either 7 days or for 28 days. So this way we could induce variation to them, because we know that the social environment is a source of variation. There could be dominance hierarchies that emerge, or there could be niche separation that all forces individuals to develop maybe different phenotypes. As a control, we have our 0-day treatment, where the fish were really on their own in their tanks. And after 7 weeks, we measured their behavior. And our expectation was that in the 0-day treatment, we won't find as much variation in the behavior as in the 28 or in the 7 days treatment. However, what we found was that in all 3 treatments, there was a lot of variation going on in these fish. So that was really fascinating. And it seems that there must be some mechanisms going on that really lead clonal fish reared in highly standardized, mostly exactly environments to develop or emerge behavioral differences. And yeah, we propose 3 different ways how this variation can emerge. The first way is they come out of the mom and are different. So that is the first possibility. So there are pre-birth mechanisms that lead to that variation. Then the next possibility is that there are some feedbacks, like I said, for example, social feedbacks or microhabitat choice feedbacks that lead to the emergence of variation. And as a third possibility, there could be developmental jumps at which time points individuals really diverge in their behavior. We don't know, but we performed an experiment, which is finished at the moment, and we write together the results. I will show you some preliminary results here. We performed an experiment where we observed the clonal fish not only after 7 weeks, but for 16 weeks from the first day of their birth, continuously. So we had a lot of small tanks in the lab with a lot of Raspberry Pis installed, which is something that Jolle Jolle's done for us as a cooperator on that study. And we observed these fish then for 16 weeks with 1 frame per second. And that produced, almost 900 hours of video at 25 frames per second that had to be analyzed. And we used a custom-made tracker for that, just to show you how that looks. The fish were in these small tanks and they were observed the whole photo period. So whenever the light was on, we observed our fish. So that could tell us when the variation among the individuals have emerged. And what you see here is, the variation was there at the beginning. Very early on, they differed. And there's no change in the variation we record over the time of the experiment. We have a high behavioral repeatability, which depicts the amount of variation that is accounted by the individual. So 50% of the variation, for example, here in the total distance swum, is due to individual differences. So let's have a look at the very first day then. What happens here? Do they emerge on the first day, or do they really are born with this high variation present? And this is, the first day of the experiment. You see the variation is present. It's not, it's not developing over the first day. It's already present when they're born. Most interestingly, we also found some maternal effects that, the offspring from some moms are much more different from the offspring of other moms. But this maternal effect could not explain the whole behavioral variation that we find here. Still, there is a lot of individual variation going on that has nothing to do with the maternal effect. Anyways, to sum this first part, natural clones are a very nice tool for us to minimize genetic variability. And we found that clonal fish differ consistently in their behavior, even when they're reared in highly standardized lab conditions. And these behavioral differences in the clonal mollies are present very early on, and there are also some maternal effects detectable. Okay, let's come back to this slide. I now focused on the individual phenotype, but the individual phenotype, also is incorporated into a larger surrounding, into the group. Most animals live together in groups, which is also part of the environment. And we now want to know what are the effects of the individual differences in the phenotype, in the behavioral phenotype, on that group living behavior, on that collective behavior. And to answer these questions, we go, for example, the way to observe groups of fish. And here's an example that Fritz Francisco, a PhD student in our group, did recently, using a very nice software that was developed in Constance with Tristan Walter and Ian Cousin, that helps us tracking the individual ID and to see what are the fish seeing, to really get the visual field of the animals. And this way we can observe what individual differences what effects the individual differences have on the group behavior. But as you see here, we can't really control what's going on in the group. We can just observe. But we want to control at least some parts of this collective behavior inside the group in order to manipulate it. And here comes the biomimetic robot, our Robofish, into play. You see me here in a video that was recently broadcasted at ZDF Terra X. And it shows our little guy, our little Robofish, which resembles a live guppy or a live molly. So it also works with a lot of different species. I will explain a little bit in detail. At the moment, you see the single Robofish, in the back a live big fish. You see that there is a robot unit below the tank. I will show that later on. And you see how the robot can integrate into a swarm of live guppies. So these fish-like robots are not, something special to Berlin. There are a lot of examples worldwide where labs use fish-like robots to study animal behavior. For example, in New York, there is a robot that resembles live zebrafish. In Italy, they developed a robot that resembles Betta splendens, Siamese fighting fish. Then we have our guppy-like robot, and also you and Constance have a biomimetic robot, which resembles more a koi carp or a larger fish. If you want to know more about animal-robot interactions and biomimetic robots, I highly recommend this review paper by our Italian colleagues. And how do these fish-like robots work? So as you saw in the middle, this is how our robot in Berlin works. We have a dummy fish, which is a 3D printed replica of a live fish that is attached with a magnet to a robot unit that is running below the tank. This way, the robot unit can't get wet, so which is highly beneficial for any of these techniques. However, we can only steer our robot in 2 dimensions, so it can't really change the height. You can change the height when you attach your dummy fish to a robot unit above the tank. Then you really can go down and move in 3D dimensions in the tank. But on the other side, you have always a robot unit on top, which might not look very very safe for the fish. So most often when fish see something above the water surface, they think it's something dangerous, like a bird or anything else that will hunt on them. So that is the downside of this technique. And as I told you, the robot in Constance, everything, the whole robot unit is put inside the robot, which makes it bigger, but also allows much better and much more degrees of freedom in the movement. So you can do much better movements then, and you can study, as it was done with that robot, how the movement of the fins and the whole body affect other individuals in a group, in a pair, in a swarm. So what can these robots do? We recently reviewed that in a small paper led by Tim Landgraf, who is also developing the robotic fish with us. And basically, there are two modes how the robots can be controlled. The first is an open loop control, which means that we can define what the robot is doing, and the robot then executes these behaviors. And then the closed loop control, which means the robot is interactive. Here's an example of an open loop behavior, which resembles a strong leader. So the robot is just driving on a zigzag trajectory through the tank. It's not reacting towards a live fish, which is, in this case, following very close to the robot, but the robot is not interacting or is not adapting its behavior towards a live fish. It's just swimming the predefined zigzag. What can you do with that? You can provide live animals always with the same social cues. So on the left side, you see the tracks of the robots, and on the right side, you see the tracks of live fish. And all the live fish represented with the same social stimuli. So with the robot performing the same behavior. And then you can really see what differences in the social responsiveness or the sociality per se these live fish have. So you see here, some live fish followed very close the robot's trajectory, while others did not follow or just moved on their own. So when you repeat these tests with the same individuals a couple of times, you will find that some individuals always follow and some individuals always not following. Okay, but what means closed loop behavior? Here we programmed the robot to follow the live fish very close. I think it's 10 centimeters. The live fish should be away, and the robot just swims behind the live fish. So it's a very weak leader. It's a follower. And this has been done by having a live tracking system installed that always feed back the position of the fish into the robot software that then calculates the next step where the robot should move. We use that in a study with Yolle Yolles to look at what are the effects of different live fish speeds on the performance of these pairs when one part of the pair, the robot, is just following what the live fish is doing. And we found that the speed of the leader then has a very strong effect on the distances among the individuals and also on the polarization. So the speed of one fish, and here we really know which fish has the effect. It's a live fish because the robot just followed. The speed of the leader really affects collective patterns, which is something that could confirm also previous research and now pinpoints these effects. But we can also merge the open and the closed loop behavior of the robot. Here the robot, for example, is programmed to swim to random trajectories, to random points in the tank, but only when the live fish is following. When the live fish is not following, like it was the case here, the robot will come back and try to convince the live fish, more or less, to follow it. And after this, one or two approaches, we can now increase the speed of the video a little bit because a live fish is now convinced to follow the robot for 10 minutes And the robot is always picking new random goal points in the tank while the fish stays close. If the fish would have left the robot would have driven back and would stay next to the live fish for a longer period and then try to execute the leading behavior. So this then produces patterns like this. Here you see the distance among the fish, and in green, phases where the robot is leading. And in orange, the phases where the robot is coming back following the live fish. So whenever the live fish was not following the robot came back to a certain distance, then stayed there at this distance for a while, for a couple of seconds, and then tried to execute the leading behavior. Sometimes it works and sometimes it doesn't work. So for example, here and here and here, it really worked. The robot swam through the tank and the live fish stayed close. But then, the live fish left and the robot came back, trying to recruit the live fish to follow again. As I said before, we find individual differences in the tendency to follow the robot. And we also see these individual differences when we have an interactive robot. It's not just the zigzag pattern. It's also, per se, a thing that the fish have, a social tendency. So the sociability that the fish differ among each other. Our aim now is to minimize these individual influences of the followers' differences in behavior on the leadership success of the robot. And this is why we use the robot as a very sophisticated leader, having these two behaviors that are described in the video before. Let me explain you. At first, when the robot is approaching the live fish, it follows well-known rules that Ian Cousin and Jens Krause developed 20 years ago now. And this zone model, you're having a repulsion zone where when the live fish is too close, the robot will swim away, having an optimal distance zone where the robot wants to have the live fish, and an attraction zone where when the live fish is there, the robot approaches the live fish. And after having the live fish around the robot for a couple of seconds, the robot switched to the leading mode. And then these optimal distance and the repulsion zones become obsolete. We only have now the leading zone, which is a radius that the live fish is allowed to go away from the robot. Having a small leadership zone, a small radius, then the robot will always come back when the fish is out of that zone. Having a large leadership zone, then the live fish has a longer way to go or to swim away, is allowed to go more away from the robot before the robot turns. So having, for example, a leading zone that is as large as a tank, the robot won't react to the live fish. It will just swim to the goal area. And this way, we can manipulate the social orientation and the social responsiveness of the robot leader. Having a small leading zone, the robot will come back very quickly when the live fish is swimming away. Having a large leading zone, the robot will let the live fish swim away for very long distances before coming back and trying to recruit it again. And this was inspired by a work from Ian Cousin and Christos Ioannou that showed that leaders differ in their goal orientation, and that this differences in the goal orientation really affects whether they are followed or not. So leaders that go directly to their goal face a high risk of not being followed, but they arrive really fast at their goals, but maybe alone. And we wanted at first to reproduce this pattern, having a more controlled design with the robot leader that we really can say how it should behave, whether it should go more to the goal or being more responsive to the conspecifics in the group. And here you see some videos. On the left side, you see a very small leading zone, meaning that the robot is, meaning that the live fish is not allowed to swim away from the robot. The robot will always come back. In the middle and then on the right side, you see larger leading zones. On the right side, it's a leading zone of 72 centimeters, which is the whole tank. And you see the robot just swam towards the goal without reacting to the live fish. In the middle, you see the robot had to come back once to the live fish to recruit it again, and then it swam to the goal area. But as you saw, for the very small leading zone, for the highly responsive and less goal-orientated leader, it took forever to reach the goal area. And that, yeah, we can, of course, measure. And we find that the time to the goal area is really bound to this social responsiveness as reflected through the leading zones. So having a small leading zone means that you take forever to reach the goal because you always have to follow your group mates. At the same time, a small leading zone means that you're very close to your soulmate, to your partner, while when the leading zone is large, you reach your goal fast, but the distance to your soulmates is also larger. And looking at the variation of these patterns, we can find a very interesting pattern that, for example, when the leading zone is small, the follower controls the accuracy. So the live fish controls whether they reach the goal or not when the robot is just programmed to follow. But when the robot is programmed to approach its goal very directly and not really look for the live fish, then the follower controls the cohesion. It can decide whether to follow or not. But in the intermediate responsiveness range, intermediate leading zones, that means that the leader can control both the accuracy and the cohesion of the group. By changing the parameters here, we can also, manipulate groups of animals then with these robots in a way how we want it. When we want to have, the group being very cohesive, then we can change the leading zone accordingly, and it will work with most of the individuals, most of the live individuals, regardless of their behavioral phenotype. Okay, so at the end of my talk, just the second take-home message. Biomimetic robots enable us to control individuals in groups. This is something, for example, that you and Constance try with your virtual realities as well. And any computer animation that is used as a stimuli, as a social stimuli for animals, has the same job to do. And we go this way, having a real robot inside the tank and not a virtual animal that the live animals can interact. But the aim is the same. We want to control the social environment. We want to get control about individuals in groups. We can pinpoint with an open loop behavior of the robot individual differences in the social behavior of our live fish. With the interactive Robofish, we can reproduce the trade-off between a goal orientation and the social orientation. And we find that intermediate social orientation or intermediate goal orientation of the leader minimizes the influences of followers' differences on the leading success, which will help us a lot, for example, also in designing autonomous driving cars or other situations where we want to have an artificial leader integrated into groups of live or other artificial agents. So my whole research is basically expressed in that kind of picture. You see some people, and many of you might know some of these guys you see here in a harsh working situation. A huge discussion about scientific theories is going on here. But what the point is that it brings together people from different disciplines. So merging individual differences on expertise and knowledge, which is very important to do experiments with the robot, doing experiments with animals that have a genetic part that has to be analyzed, animals like the clones, where we have to look at behavior, we have to look at genes, we have to look at developmental processes. So you have to combine a lot of people from different disciplines to answer the questions at hand. And yeah, this picture nicely depicts this. It was recorded a couple of years ago in Mexico, where I was with Alex Jordan, which is here, and Jens Krause. And yeah, this is Pavel Romanchuk and Joe McColeman, and here Jeffrey Mazoe and Colin Tony is also here. So yeah, I'm at the end of my talk, and I hope you enjoyed a little bit, and I hope I didn't do it too bad. So I'm allowed to maybe come and visit your facilities. And yeah, thank you very much. And a lot of thank you also to the PhD students that work in our lab with the clones, with the robots. This is Juliana and Lea and Fritz, who is, I think, also in the audience today. Okay, thank you.
All right, thank you, David, for that wonderful talk. Again, anybody who has a question, please write a Q in the chat and we'll get to you in the order. I could start us off with a question about one of the very first results that you showed, which was the experiment showing that there are inter-individual differences very, very, very early on in the life stage. And I was wondering if you could elaborate a little bit more on what mechanisms might be at play for generating those differences so early on in the life stage, because I know females can often generate variation within their broods by as a form of bet hedging, by say supplying certain eggs with more androgens and others with less. So I was wondering what you what thoughts you had on that.
That's a very good point. And yeah, at the moment we don't know the exact mechanism. So bet hatching is of course one mechanism, but that would need the females also to have some kind of provisioning. So these fish are live-bearing, like guppies or swordtails or platyfish, but they don't. The moms don't provide a lot of nutrition or any exchange to the eggs. So there are other species in that family where provisioning is strongly developed, but not in this species. So it could be, but the mechanism is not known at the moment. On the other side, it could be that the position inside the belly of the female could have an effect on that. We don't know, and I have no idea how to measure that. So maybe with X-rays before birth or so, but you don't know whether they change the position in the belly. So the other thing is it could be just a random thing, because it's so complex. The interaction of so many genes that are present is so complex that it's just a random thing whether you develop to that or to that behavior. We will go on with Professor Manfred Schartel to look at the gene expression rates of these fish at different points in time to see when we find some variation that we can link to the behavior. But yeah, at the moment we don't know.
Okay, thank you. Yeah, we have a question from Oliver.
Yeah, I have a question, but first, thanks for the great talk. Very interesting. So from what I saw, you always try to carefully avoid that fish physically interact. Like whenever the real fish comes close, your robot fish goes away. So do you have any ideas what happens if they touch? Do they then change their behavior? Is it something they don't care about, or do they then find out it's not real, they don't follow it anymore, or so? What happens?
So they occasionally touch the robot, and we can't avoid that. Even if we program in some kind of these repulsion zones or avoidance of the live fish, sometimes the live fish just makes a jump and then it touches. But we haven't seen that this is affecting their behavior very, very much. So, but maybe there is a funny story about that. There was a robot that developed, I think, in France, that was developed to lead chicken, so small chicken, and it at first overruled some of the chicken because they were too small and below the sensors. So they really had to take care of that to avoid these losses. For the kind of robot that we use, I think this is not so important. We don't see that they change their behavior, and we can't hurt the fish with our robot. So it's something more or less normal that could occur also among live fish.
No, my question was more like maybe the real fish would then find out it's like something weird. It's not another real fish, and would maybe change their following behavior, because otherwise you could think of, well, if it's not like a fish-like shape, but anything else that moves, maybe fish would also follow.
Yeah, for us, it was a very interesting discovery that we found out when we just painted the eyes on the dummy. It didn't work. So they didn't accept the dummy fish. But then we glued little glass eyes made for toy teddy bears. Mm-hmm. And then it worked. And you could really place this replica inside a tank of guppies, and the males start their sigmoid courtship displays to that dummy. So it's more or less like a key cue like Tinbergen proposed, a social releaser that is bound to the eyes and maybe the shining of the eyes that is important for this kind of species. So it might be that different species react very differently. For example, many other species use chemical communication, and they might smell it's not a conspecific. But for the guppies and for the mollies, it seems as it's not super important that it has to smell like a conspecific.
Our next question is from Liang.
Very nice talk. I have maybe two small questions. The first one is, I'm wondering, when you run experiments with real fish interacting with robotic fish, did you find some situation like the real fish will react to the robot at the beginning very, very frequently, very, very smoothly, and as time goes on, then the real fish might lose interest to the robotic fish and then interact with the robotic fish less and less. Did you find this, or?
Yeah, we find that and published that in the study I showed you on the social responsiveness, that the strongest following responses we get when the fish is new to the environment, so new to the tank, new to the robot, and we find the same habituation to the situation when we test the live fish with other live fish. So we can't really disentangle whether it's losing the interest in the robot, or whether it's just getting more familiar with the tank environment, with the whole situation. Because our guppies are not always shoaling, so they're facultative shoaling. They only come together very close when they're in a risky environment, an environment they don't know, where the predator could be in every corner. And when it's safe and they are familiar with the environment, they go on their own, even when they're in a group surrounded by other live fish. So it might be very different to zebrafish or other species that are more shoaling. So herring or really shoaling species that they show much different behavior. But at the moment, we see that losing interest, but they show the same losing interest also when they are tested with or interacting with other live fish. So we can't really think it's a robot. It's a species-specific feature that we have here.
Okay, so you compared the interacting behavior to the real fish and to the robotic fish there. Okay, wow, this is really cool. My second very brief question is, can you briefly describe the future direction of this project relative to robotic fish? Like in your talk, you didn't mention what's your future direction, what's your planning to do with your robotic fish in the future?
So that is a question. That is a very, very good question. And we get these questions very often, and the answer may differ whether you ask me or, for example, Tim Landgraf, who developed the robot. So from the engineering part, the idea is clearly to put much more intelligence into the robot, to put in deep learning algorithms inside the robot, so the robot can adapt to different individuals, different species on the run, so that you just place the robot inside a tank and then it learns how to deal with certain behavioral types with certain species on its own. So as an information gathering and information processing unit, more or less. So for me, as a biologist, I would say we want to use it to test theoretical assumptions and theories that we or other people developed, and then having a system where we can put exactly these theories in and test whether they work in the real world, whether they work with fish, whether they work with different environments, with different complexity, structural complexity in the environment. And this is the way I would go with that program or with that project. So, but yeah, we always have to find together then with the engineers that want to build much better robots and us biologists that want to test much more difficult or complex theoretical models.
Cool, thanks. Yeah, this is the bio inspiration and the bio understanding. Yeah, thanks.
Do we have anybody else who'd like to ask a question?
Yeah, hi. Great talk, Gain. Very interesting questions, both the individual variation as well as the leadership with, robots. So my question was, kind of building on what Liang was talking about in the sense of how the responsiveness of these live fish to robots can change over time. I was wondering, okay, so one way is to manipulate the leading zones. So if you have a moderate leading zone, you strike a balance. But for example, if there are some individuals which are low in social responsiveness, would they require something like building a relationship, like having a lower leading zone so that they first kind of make sure that that loop is solidified and then they sort of lead them in? And could this also be in theory done through something like having the fish lead them to a reward so that later trials would sort of, concretize that relationship or make them lead them consistently?
Yeah, that's a very good question. And for sure, the approaching a live fish will change how the live fish will react on future approaches. And some very, let's say, shy fish. I know that these are anthropomorphisms, but let's say it's a very shy fish. You have to approach very slowly then to convince it to follow. And we tried this with the robot, having the robot adapting to the reaction of the live fish. So when the live fish swam away, when the robot approached the next time more carefully. And this really makes a difference. So then a live fish that swims away first is then approached more carefully, and then it is better convinced to follow in the end. So this is out as a preprint, I think, and Tim Landgraf led this research. So having, not only an interactive robot, but also a robot that can adapt its own rules on the run, can help them to circumvent, these individual differences or the problems that arise when you always approach with the same rules.
Great, yeah, I'll check that preprint. Sounds good.
Okay, well, with that, if anybody would like to stick around a bit longer, you're welcome to do so. Otherwise, we will see you all next time.